to understanding folding mechanisms. Thinking a little bit harder about mechanisms, we can envision a conformational energy landscape that helps us describe the folding reaction. And in this cartoon, we have this three-dimensional plot. Really, we'd have to remember that the folding reaction is much higher dimensionality. But let's think of the x-y axes here as the degrees of freedom that are available to the chain. And then the free energy is in the z-axis, right? So we start out in the unfolded state with a high number of degrees of freedom, so above the rim of this funnel, right? The unfolded state can adopt a very large number of high energy conformations. But then as we change the conditions to allow for the protein to fold, then we restrict the number of conformations that are available, and ultimately we find ourselves deep in this funnel where the low energy state, the native state, is the only one that's available. So unfolded conformations are isoenergetic and high energy and unstable. The folded state is at an energy minimum, and the landscape defines a topography. So we can talk about the energy landscape. So what's the topography? Well, we might have multiple minima. So that you could imagine as describing perhaps an intermediate or a pathway. Maybe we have to go through this intermediate and we have another barrier to overcome to get from the intermediate to the native state. So this is a useful sort of cartoon, right, model system for understanding the way that proteins fold and for understanding the way that proteins misfold. So we could have a rough landscape in which a protein has a difficult time finding its way to the native state. It means there are multiple minima and multiple barriers. Those non-native structures might also be at very low energy minima, meaning that they're can be quite stable relative to the native state, and therefore populated to an appreciable extent. So the landscape allows us to sort of conceptualize some of the problems and issues in protein folding, such as amyloidogenesis. Now we're going to come back to a description of amyloidogenesis at the end of this lecture, but it's worth mentioning here that misfolding is an important part of protein folding, and frequently many diseases result from the misfolding of proteins and the formation of non-native, right, alternative structures that are in competition with the native state. Another detail that gives us a general sense of the rules of protein folding that we can deduce from our kinetics and equilibrium experiments is that structure determines folding rates. So I said that proteins tend to form these nuclei of structures around which the rest of the protein structure accretes. But there are more basic rules that emerge. First of all, well, let's just look at this figure for a second. So we're looking here on the y-axis at the log of the folding rate versus contact order. What's contact order? Well, simply stated, it's just a metric of the relative contribution of different kinds of short versus long range contacts. So a low contact order protein is a protein with a very simple topology, one whose structure is predominantly determined by local contacts, versus a large contact order protein, which is a protein that's topologically complex and whose structure is mostly determined by long-range contacts. So when we look at all of these proteins that seem to fold according to a two-state model, that is to say, without appreciable intermediates, we see this very interesting behavior emerge. Proteins that fold fast tend to be low contact order, right? And now we're not talking specifically about any particular kind of structure except to say that it's short range. Proteins that fold slowly tend to have a more complicated topology, more long-range structure. So the very, very simple relationship between the distance dependence of your fold, right, and the rate of folding. So long-range contacts or long-range order makes the protein fold very, very slowly. That's a powerful observation. Remember that this is without respect to any kind of particular structure. So earlier I said that each folding pathway is unique, Each folding reaction is unique. Maybe a different kind of intermediate is populated. Maybe the transition state has a very different structure from one protein to the next. But in this case, we say that the rules are very simple and don't depend on the detailed organization of the protein at all. It's simply the matter of how close or long range the overall structure of the protein is. Very powerful.